The overall goal of this procedure is to construct an in vitro lung tissue model for studies of M. tuberculosis infection. This is accomplished by first layering a fibroblast collagen matrix onto the filter of a cell culture plate insert. In the second step, infected immune and lung epithelial cells are seeded onto the fibroblast collagen layer. The resulting 3D tissue model is then air exposed to facilitate the mucus secretion and stratification of the epithelium. In the final step, the tissue is harvested and mounted. Ultimately, immunofluorescence microscopy is used to analyze the formation of M. tuberculosis-induced early granuloma.
The main advantage of this technique over existing methods such as in vitro monolayer of cells is that the cells in this system are present in a 3D microenvironment, allowing the formation of granulomas upon infection with virulent mycobacteria resembling human TB.
To prepare collagen-embedded fibroblasts, begin by treating lung fibroblast cells with trypsin for 10 minutes at 37 degrees Celsius. When the cells have detached, stop the reaction with 8 milliliters of complete DMEM and then spin down the cells. Resuspend the pellet at 230,000 cells per milliliter in fresh DMEM and place the cells on ice. Next, add 1 insert per well into a 6-well plate and dispense 1 milliliter of freshly prepared acellular layer mixture into each insert, making sure that the mixture covers the entire insert without